He's from the University Victoria in Canada. And today we are very pleased to welcome Dr. Shafiq. He's a researcher at the Institute of Neuropathology at the University Medical Center in Hamburg, in Germany, working in the lab of Professor Markus Glatzel. And he completed his PhD at University Medical Center Göttingen in Germany, where he studied protein misfolding in rapidly progressive Alzheimer's disease. His current research focuses on extracellular vesicles in neurodegeneration, and he will talk a little bit more about it. So thank you again for joining us today, Dr. Shafiq. So the floor is yours.
So thank you very much. First of all, thank you very much for the invitation, and I'm glad to be presenting an SNEV seminar, so to speak. Thank you for the kind introduction as well. And so I think we jump right in into the presentation, and the topic that we will be talking about is actually what do EVs do in the Alzheimer's disease? And we will discuss at least two aspects of the disease and the EV interaction to the disease. One of them is going to be the plaque pathology, and the other one is going to be the neural network function. So I assume that the audience is quite diverse, so I tried to keep the things simple and straightforward. I always like to start with this figure from the review from Bonnie. And we all know now that the EVs, extracellular vesicles, they are very robust cargo messengers. They are composed of signature molecules like protein, DNA, RNA, which depends on their source. And henceforth, they can be involved in trophic signaling, paracrine signaling, autocrine signaling, and they have their own disease-related signatures also, like oncosignals, infection signals, and whatsoever. Now, particularly in the nervous system, of course, everything remains true. They are responsible for doing the transportation because of their certain signature makeup. However, in case of the neurons, we published last year that they also carry some receptor binding proteins, which can be actually responsible for eliciting some signaling mechanisms in the neurons, which is also interesting. And as we are discussing about Alzheimer's disease, I will just give small pointers. First of all, it's one of the major dementia. There are currently between 35 million to 40 million people affected, and this number is supposed to be double by the year 2050 by the statistics of National Institute of Aging. And Alzheimer's is not a simple disease. It's a very simple slide to talk about Alzheimer's disease. Actually, Alzheimer's disease is a syndrome, so it contains multiple abnormalities, particularly, of course, clinically speaking, progressive cognitive decline is one of them. Then, of course, there is a loss of neurons, loss of brain mass, whatever you can say. And of course, this goes hand in hand with the inflammation and immune activation. There is profound mitochondrial dysfunction also. And one of the features, which is actually common in all the neurodegenerative diseases, nearly all the neurodegenerative diseases, is protein misfolding. And when we talk about AD, in AD, the two major proteinopathic proteins are amyloid-β and tau, which are also the neuropathological hallmarks of the disease. So if you see that the neurofibrillary tangles, after the tau gets hyperphosphorylated, it clumps and it makes these particular triangular structures or pyramidal structures, you can say. And these are normally exhibited as neurofibrillary tangles. And on the other hand, amyloid-β starts clumping and it starts making these plaques. There is a variety of plaques. Majorly, you can say that there are non-compact diffuse plaques, and then on the other hand, there are compact core plaques also. So these would be two basic forms. In my talk, the first part of the talk, let's say, I would be focusing more on the amyloid-β plaques. So we'll jump into it. the subtitle would be Extracellular Vesicles Modulate the Amyloid-β aggregation in Alzheimer's disease. And I'm going to speak in support of this particular idea. We have a very sparse literature when we talk about the interplay of extracellular vesicles, particularly related to amyloid-β plaque pathology or plaque formation. Of course, there are definitely a lot of studies discussing about EVs coming from Alzheimer's disease, their proteomic makeup, their lipidomic makeup, their cell of origin signature. And of course, there are many efforts going on in regards to the biomarker establishment of the EVs or based on EVs for AD particularly. But when we talk about the mechanistic studies, which highlight the effect of the extracellular vesicles with amyloid-β, they're not so much. And one of them is actually coming from 2006. It's quite old in that aspect. And they're the authors, Rajendran, basically, they explained in PNAS, this paper was published, that the amyloid-β has ALEX associated with it, which somehow they concluded that it could be the exosomes which are entrapped in the plaques. Then my group or our group has published also something supporting to the idea in 2016, where we showed that the extracellular vesicles, they do derive fibrillation. And then rather recently, not so much, in 2024, there was a study where they did cryo-electron tomography of the amyloid-β plaque from Alzheimer's disease brain. And they also saw some lamellar bodies in the plaque along with the fibrils, you can see. And they also denominated that this could be actually extracellular vesicles. So we started our quest with the biochemistry, let's say. We know that the amyloid-β is basically a cleavage product of a bigger protein, which is called as amyloid precursor protein. There are two cleavages involved, one done by base 1, and the other one is gamma secretase complex. And then it results in a smaller peptide, which ranges from 38 amino acids to 42 amino acids, mostly. And this is called as amyloid-β. And the most toxic form is considered to be the amyloid-β 42. And as I said, no, it's a cleavage product. It starts this journey as a monomer. But biochemically speaking, this monomer is not thermodynamically stable. So what happens is that it starts to clump together. This also happens in vitro. So if you have a solution of a recombinant amyloid-β, it would start to make complexes and oligomers. It also happens in the physiological context, so in vivo, of course. And as you can see, that there's a very simple graphical explanation. So if you follow this red line, you see that this is actually representing the toxicity. So the toxicity is the highest when you talk about the oligomers. And these oligomers are then conformed or clumped together to the protofibrils and ultimately to the fibrils, which constitute the plaques, and which are supposedly relatively inert in comparison to the oligomers in terms of their toxicity. And we thought, okay, this is happening, and there are multiple receptors also of amyloid-β, which are actually deriving the toxicity in terms of the neurons. Would it be possible that one of these receptors is also present on the surface of extracellular vesicles? And this also allows to bind the extracellular vesicles to the amyloid-β monomers or oligomers, and then henceforth provide a platform for sequestering or for amyloid-β formation or plaque formation, let's say. This was the basic hypothesis. And why did we think about it? We basically have, as a group, we have a history of working on the prion diseases and also a biochemical aspect of cellular prion protein. And cellular prion protein is actually one of the potent receptors for amyloid-β, oligomers in particular. And if this interaction takes place on the surface of cell, of course, what happens is that it somehow organizes the axis for fin signaling, and it ends up in the neurotoxicity. But PrPC, or cellular form of the prion protein, is not only present on the surface of the neurons. What happens is that the N-terminal is a pointer, sorry. Yeah. So the protein is basically a longer 200 nanometer amino acid protein, and it has a flexible N-terminal domain, you can see here, and a C-terminal globular domain. What happens is that it is possible, there are some enzymes which cleave the N-terminal from the protein, resulting in the N fragment or N-terminal fragment, which has the binding sites for the amyloid-beta oligomers. And on the contrary, what also happens is that ADAM10 mediated shedding also takes place on the surface of the cells, and that ends up in the release of extracellular relatively complete molecule of prion protein. And this is also in turn capable of binding amyloid-beta oligomers. And in the third place, what happens is that, of course, there are extracellular vesicles, which are composed of membranes, and most of them are actually cholesterol-rich domains, and prion protein is associated to those domains. So it ends up in a high abundance in extracellular vesicles, particularly in case of the neurons. So if you talk about these, all these forms of prion protein are present in the extracellular. So what extracellular milieu or extracellular environment, what happens then that they could actually act as the vacuum cleaner, let's say, for the amyloid-beta oligomers. So they can actually bind to the oligomers and reduce toxic burden. This is what we were thinking, or we think. And then, of course, now for the hypothesis, of course, what we wanted to see, we wanted to highlight, or we wanted to focus more on the extracellular vesicle and their presence in the Alzheimer's brain, or how does this interact to the amyloid beta? For studying this in particular, we had three aims, let's say. First, explore the EVs from the amyloid beta or Alzheimer's disease brain, and explore the plaques. What is their biochemical composition, and do they have prion protein in them, and also if they have EV markers in them. EV markers have been shown previously also. And then also what we wanted to see that if this EV surface expressed prion protein is indeed involved in the amyloid aggregation. So we started with the isolation of extracellular vesicles from the Alzheimer's disease brain tissue. And typically, I would want to discuss a little bit something. Typically, for the EV isolation, what do? We cut the tissue in a suitable medium, and we put some collagenase or papain or anything, any protease that would help us to release the extracellular matrix, let's say, and also EVs in turn. But we noticed that if we put collagenase for the EV isolation, what happens is that it also cleaves our protein of interest, as you can see here. So we have, let's say, all the classical way of isolating, like we cut the tissue and then we do some pre-clearing. Then we end up doing OptiPrep, and from the OptiPrep, we get first two fractions where we have majority of extracellular vesicles. And from them, we wash them with PBS, and after washing, of course, this is what we have. So for example, if we use collagenase, this 0.5 milligrams per ml is actually the concentration of collagenase. You see that we have cleavages happening in our protein of interest, which is prion protein. And if you see the normal form of the prion protein is a tad bit higher, this also happens here in the fraction 2 EVs. And if you see, there is a shift in the molecular weight. And this was one of our hindrance in using the proteases in our protocol. The same thing you can also see very pronouncedly here with CD81. If you use collagenase, what happens is that CD81 is getting cleaved, and you have a certain secondary band of the CD81 whenever you have collagenase. So we opted for, or we actually optimized another protocol where we did not use the protease of any sort, no collagenase, no papain, nothing against collagen or anything. So, and then we ended up having actually quite decent amount of EVs to proceed further. And what did we do? We used the same protocol, and in the cohort of Alzheimer's disease patients and control autopsy brains, we isolated the EVs. And after isolation of the EVs, you can see that the EVs are very nice here. Even in the AD EVs, I should have put maybe a bigger figure here, where you can also see these thin strands. These thin strands are actually amyloid beta strands or amyloid beta fibril. So we were excited to see them as well, that they could also be found in the same density fraction as the extracellular vesicle. Of course, I'm not suggesting that they are attached to extracellular vesicle. This could only be, of course, the effect of the optic gradient that they end up in the same density. But then what did we do? We did some proteomic analysis in order to check if they express the classical EV markers or not. So we, of course, have a battery of markers here, or the proteins which are associated normally to the extracellular vesicles. And then what did we do? We also checked for the expression of different proteins of interest, among them prion protein, cellular prion protein. And of course, the expression of the prion protein or cellular form of the prion protein was higher in case of Alzheimer's disease patients compared to the controls and even the early stage Alzheimer's patients. So it sets us in the same direction as we were thinking, that of course it could be that in the late stages of Alzheimer's disease, there is a higher expression of the prion protein on the surface of extracellular vesicles, which would also contribute to the sequestering of the amyloid beta oligomers into plaques. Then we confirmed it also. How did we confirm it? Of course, we did use only CD81 as a marker for extracellular vesicles. And we have a look or have high magnification pictograph of the amyloid beta plaques. And in the plaques, you see that there are signals, as have been suggested by previous studies also, for the CD81. But what we also saw that these signals for the CD81 were actually quite close to what we observed for the PrPC. So cellular prion protein or cellular form of the prion protein was also seen abundantly, relatively, in the core plaques particularly. I'm not talking about the diffuse plaques, it's different, but core plaques in particular. So suggesting us again that it could also be associated like, okay, that the EVs with the PrPC on them provide a seed for the clumping of the amyloid beta plaques. Then as we did the proteomics already, we tried to see the proteomic signature or biochemical signature. Does this go in the favor of our hypothesis or not? So what you see here is a correlation matrix for EV proteome from Alzheimer's disease and also from control. And when you see that the EVs, of course, there were, I think, around 4,000 proteins which were detected. And of course, not all of them are positively or negatively correlated to the PrP. And whatever was correlated to the PrP or the prion protein, cellular form of the prion protein, that was involved in multiple different things in Alzheimer's disease, which was different for the control EVs. So you can see that in here is ER signaling, synapse regulation, autophagy, and everything involved here. In case of controls, the EVs were, or the proteins which were directly correlated to the prion protein cluster, were associated to the cytoskeleton, metabolism, neuronal development, in the general gene ontology terms. But particularly for Alzheimer's, the proteins which were only positively associated to the prion protein, they were involved in the amyloid beta clearance pathways, which is again suggesting us, okay, these proteins, along with so PrP is involved with certain machinery which is involved in the clumping or amyloid clearance pathways. So then we further wanted to confirm it in the mouse model. because this is something which has not been shown, or this has not been in detail done by any other group. So what we did is that we wanted to check the other way around. So first we approached the EVs. Now we wanted to approach the amyloid-beta plaques, actually. And in here, we are using 5xFAD mice, which has different background for prion protein genes. So it could be wild type. It was also in, so we had three groups: wild type prion protein background, hemizygous prion protein background, and knockout prion protein background. And what did we do? As you can see here, we did super classical biochemistry. We used a series of buffers along with differential centrifugations and isolated, for example, soluble proteins from the tissue, the proteins which are associated to the membranes from the same tissue, using a harsher buffer, let's say RIPA buffer. And from here, whatever was in the pellet of the second centrifugation, that is usually containing, so to speak, insoluble proteins, which are, as they are not soluble in the RIPA buffer. And RIPA buffer is the buffer which we normally use for homogenizing tissues, running Western blot, and whatsoever. So what did we do? We took this pellet, which would definitely also include the amyloid plaques, because they are not soluble in RIPA buffer to a major extent. And then we dissolve them in the urea, thiourea. So it's a super strong buffer. It's a 7 molar urea, 2 molar thiourea, 4% chaps. So it's a buffer that dissolves nearly everything. So we use this actually and dissolve the pellets. And after dissolving the pellet, so to speak, insoluble proteins, we did some biochemistry. And from that biochemistry, the blots do not look very nice because of the buffer. It's a very strong buffer, but because of the certain higher concentration of urea, thiourea, you end up seeing very disorganized sort of bands. But anyhow, if you see, the first group is actually 5xFAD, so amyloid model, along with the background of wild type prion protein. And the second group is hemizygous, and the third group is a knockout here. And if you see in the wild type background, you see that there's, of course, a higher PrPC, but also there is a higher concentration of CD81, which is a marker for the EVs. And we are talking about the plaque-associated proteins, all the proteins which we get from the fraction where we dissolved plaques. And if you see the plotulin also had relatively higher abundance in case of wild type background, and likewise Alex also had higher levels in the case of wild type prion protein background and hemizygous. We also confirmed it using expansion microscopy. Sorry. And what we did is, of course, now we are staining it with amyloid beta and CD81, of course. And you can see wherever we have positive or prion wild type background from the tissues, we have higher accumulation of CD81 in the plaques. And this was not the case for the knockouts brains, as we see, or plaques from the knockout brain. Again, strengthening our hypothesis that the presence of the prion protein is actually allowing the sequestration of the amyloid beta onto the EVs, or let's say is allowing the hybridization of the EVs to the amyloid beta plaques. And then furthermore, we wanted to then see some structural details. So we wanted to discuss what's going on biochemically on the surface of extracellular vesicles itself. So for that, what did we do? We put our attention to the recombinant amyloid beta system. And as I discussed in the very beginning, that the monomers of recombinant amyloid beta, they also tend to aggregate over the time. And this is also true if you put something in there. In this case, what did we do? We added PrPC wild-type EVs or PrPC knockout EVs, and then monitored the effect of the EVs on the aggregation kinetics of the A-beta aggregation system. And how did we do that? We started with the brain-derived EVs. In this case, you can see that these EVs are coming from PrPC wild-type and PrPC knockout mice. So, and these are then isolated from brains. So they are brain-derived EVs. And what did we do? We did the assessments, NTA, and you can see electron microscopy. And then we did some super resolution microscopy after incubating those EVs with the amyloid-beta. And here you can see that the EVs are labeled in green, and amyloid-beta is conjugated with the star red dye, and this is STED microscopy. And you can see that after the 60 minutes of incubation, you already see a lot of clumping happening in case of the wild types. If you add wild type EVs into the amyloid-beta system, you see aggregation happening instantly. This, however, is not the case for the knockout. It does catch up after 24 hours. You do see some clumps, definitely. But of course, the extent of the clumping is much higher and much faster in case of the wild type EVs incubated with the amyloid beta. And then to further, in the hope of obtaining molecular structure, we also use a method which is called as synchrotron small angle X-ray scattering. It's I don't think that most of the people are familiar with it. It's the biggest microscope that there can be. It's the synchrotron is basically an electron ring. In Hamburg, we have one, which is, if I'm not wrong, it's around 6 kilometers in diameter or something like this. And what do? You accelerate the electrons at very high speed. And when you then decelerate them, they produce a very brilliant beam of X-ray. And this X-ray you can then use to study in solution, because what we want to see is that dynamic reaction, not the end stage reaction. So in solution, we have the same aggregation system along with or without extracellular vesicles of 2 different types. And then this is what we subjected to the synchrotron. Well, I added this just to maybe fascinate you guys, is that this yellow line is basically the synchrotron itself, the instrument that I'm talking about. And this blue building has the something which is called as experimental hutches, where you produce X-ray and then use it for the scattering experiment. And what you get out of the scattering experiment, as you can see here, is something called a circular vector. So it's basically a circular vector of the intensity of the different scattering patterns happening, and different portions or different parts of the curve actually give you the information about size of the molecule, shape of the molecule, interface, and the internal structure. And by doing so, what did we do? We got the scattering curves. It's much easier if you focus on this part of the graph, actually. So if you see that the PrPC knockout EVs were there, PrPC wild-type EVs were there, and only a beta was there. PrPC wild-type EVs, along with the amyloid beta, they start with a higher clump size or higher aggregate size, and they keep on growing also. Of course, this growth also happens in A beta alone system and also the system where you have knockouts also. But definitely, it's much more pronounced in case of amyloid beta. Then we wanted to see, okay, in the aggregation system, you see the thing happening, but what if you treat with different type of amyloid beta? So it means that they could be oligomers, they could be end-stage fibrils. Is their interaction different among each other, or is the same thing? So for this, we use Neuro2A cell-derived EVs. So this is a neuroblastoma cell line. And of course, we did some characterization and mass spectrometry for checking the expression of the different proteins associated with EVs. Finally, if you see here, what do we have here? We have, as I said, oligomers, amyloid beta oligomers. which are then incubated with the EVs coming from wild-type background. This you see here. And oligomers coming from knockout background along with the oligomers, which you see in this panel. And there is a day-night difference between the clumping or between the association of the wild-type EVs with the amyloid-beta oligomers compared to the knockout ones. And same is the case for the fibrils. If you have end stage, you do see more association of the EVs to the fibrils, not so much in case of the knockouts. However, the clumping or the overlap or the interaction of EVs is much higher for the oligomers compared to the fibrils. Then we thought, okay, let's investigate this a little bit further using cryo-electron tomography. So cryogenic electron tomography, I think most of you guys are already familiar with the technique because it's used quite extensively in case of extracellular vesicle research. And what you do is basically you vitrify by plunge freezing your sample and on a grid. And then, of course, you put the sample and then you rotate the sample while taking execution of the transmission electron microscopy. And ultimately, you put them all together in a sort of a stack, let's say, and you get a high resolution transmission electron microscopy image without any artifacts or, let's say, small amount of artifacts. And our hypothesis so far has been that, okay, amyloid beta, in this case, now I'm using the terms adults because in vitro, whatever you get from the recombinant amyloid beta, classically in literature has been referred to as adults or ADDLs. And if you put them with the wild type EVs, you definitely would assume that there is a higher interaction. And this is contrary to what would happen when you have the knockout EVs. And this is what we also saw in case of cryo-electron tomography. You can see that in case of the wild types, what happens is that wild types, indeed, the extracellular vesicles are providing a sort of a nucleus for the association. You see all this cloud is actually amyloid beta. So for the attachment of the amyloid beta onto them. And this is not so pronounced in case of the knockouts. And of course, you can also model whatever you see. And from here, what we actually modified our hypothesis a little bit, that instead of just one EV serving as a seed, there is actually a clump of EVs, and then on top of that, there is a deposition of amyloid beta happening. So along with it, we also saw some other interesting things like internal cargo. Also, there were EVs which are actually trying to have interaction or adhesion, let's say. There are some EVs, well, I could only find one extracellular vesicles out of 100 samples that I had, where I could see something similar to protofibrils. And then, of course, there are a lot of amyloid clumps. But anyways, this is something that is still pending for further investigation, let's say. But so far, regarding the interaction of extracellular vesicles to the amyloid beta plaques, what we can say that EVs definitely expressing the PrPC on the surface promote a receptor-driven amyloid beta sequestration or aggregation, which happens both in vitro, in vivo, in Alzheimer's, and in the mouse brain or mouse model brains also. So then, if you noticed, the talk had two parts. One was amyloid beta plaques, and the other one was neuronal network. I will start with the second part of the talk. It's not super extensive as the first part because we are still working on these aspects. So we thought, let's do something explorative. And what would happen if we actually treat the neurons with amyloid beta derived EVs? Of course, there is a lot of treatment studies done in the EV field in general, but not really for the amyloid beta derived EVs actually. And we of course had some idea that what we would want to do is that we would derive some iPSC neurons, so some human pluripotent stem cell derived neurons, Both from control setting and from Alzheimer setting, we will do different types of treatments with different extent. So immediate or acute treatments, and then the readout would be calcium imaging. We would do some subacute treatments where we would focus on proteomics and transcriptomics, and also long-term treatment for the same, let's say, explorations. And we started with a model. Actually, we obtained the model from Cornell Institute. We obtained the initial starter vials, let's say, and then we basically, a colleague of mine, Dr. Andreu Matamoros, he was extensively busy in reprogramming the iPSCs and, of course, their validation and bringing them to the level of the neurons. And day 80 and 85 was something where we treated the cells when we saw them, okay, they are really having a good network with each other. And this is where we treated them with EVs. EVs also coming from Alzheimer's disease and also coming from controls, as you saw previously. And in the first case, as I said, our idea was to see what happens, whatever happens in the calcium imaging, or if there is any differences in the connectivity of the cells to each other when they are treated with the EVs coming from Alzheimer's or control brain tissues. And what you can see here is a schematic explanation. Okay, the dye, you actually treat the cells with the dye, incubate it for some time, like 40 minutes, and then you give 2 washes. And then, of course, you treat the cells with the EVs and you see if there are any ionic changes happening. And the calcium, when it's released, it binds to this fluorophore dye, and then it gives an emission of 523 nanometer, which you normally observe in the spinning disk or any other modality of live imaging. So what we saw is very interesting. Here we are talking only one cell line. I would only be able to discuss one cell line. And the most important or the most signature variation that you can see is actually here, down here. If you see this, if you treat the cells with PBS, they are not showing any particular differences. But if you treat them with the controls, you see that the patterns of firing start to synchronize a little bit. And if you treat them with the sporadic AD brain-derived EVs, what happens is that they actually develop an epileptic seizure. Bam, bam, bam. So it's a very episodic thing that happens, which actually represents that there is the extent of network is only limited to few neurons which are firing together, and the overall connectivity of the whole culture is reduced. And this was very interesting to us, and this is something that could also be actually related to the behavior of the neurons in the brain of Alzheimer's disease. And then we thought, okay, what's the mechanism of it? And for that to see, we started to look at our proteome data originally from AD brain-derived EVs. And here, surprisingly, you do not see any upregulations, a high level of upregulations. What you see is, okay, tau protein, annexine, some other markers. But surprisingly, if you see in the down-regulated proteins, we found some molecules which were responsible or which were associated to cell signaling and calcium balance. Particularly, one of them is GABA-B2, and we also found GABA-B1 actually, which is also not mentioned here, but we found them. And we think it could also be that the effect of EVs, the control EVs, is actually more pronounced in terms of support of the cells than the ADEV. So it's a counterintuitive hypothesis, because what you see is that after the treatment of the ADEVs, you see that there is development of these epileptic seizures. But what I'm trying to say is that it's a sort of an inverse effect, because you see the supporting molecules like GABA-B2, GABA-B1, and there were some ATP-related molecules also, which are highly present in the control EVs. So this is an avenue which we are still focused on, we are still working on. We tried to do the proteomics of those cells, of course, after what you can see here is 5 hours and 24 hours of the treatment. And so far, we have been having some metabolic terms or the proteins which are associated to the metabolic terms, like protein synthesis, proteasomal complexes, and protein DNA complexes, something like this, but not particularly for the signaling. This is something that we are still trying to understand mechanistically. We are also looking on to the long non-coding RNAs for that in the AD-derived EVs, actually. So these could be something that are affecting the cells in a weird way, and that is something that might be leading to difference in the firing pattern. I would conclude, as I said, no, it's a very squeezed section regarding the neuronal activity. It's not a neural network activity. It's not finalized. We are still working on it. But what we can say for sure is that definitely the Alzheimer's disease-derived EVs, particularly from the brain, they do affect the activity, neuronal activity or network activity in the neurons. And as I said, we still have some avenues to discuss and to study for, and we are working on it. And one more aspect that we would be interested is actually to characterize the EVs in Alzheimer's disease subtypes. It's also very important. But with this, I would like to thank all the collaborators, particularly Andreo and Emina, my other MSc students, and also for you for your attention. Thank you very much. And of course, any questions are welcome.
Thanks so much. So we have some questions in the chat, and then I see Vic has his hand up as well, but perhaps, and I think Vic, you were the first one with the question in the chat. So if you want to unmute and ask your question, feel free to do that.
Yeah, absolutely. Thanks a bunch. Thanks for the presentation. This is super exciting. Definitely going to be reaching out afterwards because we work with cortical organoids. Really good avenues for use for this. So, have you seen any evidence for the uptake of amyloid-β in your EV conjugation experiments?
So you mean that that the EVs would bring the amyloid-β associated to them, and then the cells would actually be taking up the amyloid-β? Is what you're trying to say?
Yes, exactly.
Yeah,
yes.
Of course, this is possible. This is possible also, and we are also thinking in this particular dimension also. Could be that EVs are a very effective carrier. By adsorption of amyloid-β on the surface, they could actually bring it very effectively to the cells. But per se, we did not see anything by microscopy, I would say. But theoretically, of course, we are thinking in this direction also.
And the question would be, what would be your expected rate of lysosomal escape? Yeah. Then yeah, let's save that for an email. Thanks so much.
Thank you very much. Thank you. Yeah, actually, the voice was also not very clear.
Thanks, Vic. It sounds like he'll follow up by email. So I know you had another question in the chat as well, but I'll let you ask that by email as well. Rena has a question. If you want to unmute and ask your question yourself, you can. Otherwise, I'll just speak it out. So I'll give you a couple seconds. Okay, so the question was, what do you prefer for EV isolation? Should we be including DNases or proteases or not at all?
Well, we do use DNases, so we do include the DNases, definitely. But what we do is that we do not include any kind of protease. So maybe what you can do is actually look for the paper from us from 2024 in Jeff, and you would see in details whatever the protocol is, but definitely, so we do not rely on the how is it called, the proteases, collagenase or papain or anything, because I think it is important to have the EVs in their native structure than to have more of them. So in order to avoid any artifactual, let's say, readouts.
Yep. And then it looks like Bárbara had a question, so I'll let her ask
I
that.
can ask. Hi, thank you for the presentation. So you use this animal model with different PrPC genotypes. Do you think this model captures the rule of the PrPC and the Alzheimer disease itself? And another question, I don't know if I missed it or not, but you only use male mouse models, or also you used females in your study?
No, the mouse models were mixed, so we used both of them. No, it's not only one. And yeah, maybe you were referring to the isolation study. No, no, actually we used everywhere the both genders, because we learned our lessons from review. For our initial paper, we had only one gender, females, and then the reviewers asked, no, you have to include also the, males. And then, so from now on, not from now on, from then onwards, we always use both genders. And regarding the 5XFAD question, of course, I would not say it's an Alzheimer's model. It's an amyloid model. No, so it's there is a difference. Alzheimer's model, as I said, Alzheimer's is much more complex. It's not only amyloid beta, not only tau, it's mitochondria, it's immune regulation. So it's super complex. But of course, we wanted to dissect the process, let's say, and we wanted to study a certain aspect which is related to amyloid beta. So that's why we included only 5XFAD. However, I didn't show some data here. We also used another amyloid model, which is called as APPPS1 model. I think if you're from the neurodegeneration field, so you would know it. We have also different variations of the expression there, like we have knockout hemizygous, homozygous wild type, and then overexpressing TGA20. So, and there we also could see the same results. They were not as nicely oriented or as nicely made up in the figure, so to be included in the presentation though. But the message remains the same. So yeah.
Okay, thank you.
And just to follow up on that, I guess, did you look at any sex differences in the models?
In the models, we actually didn't find any sex differences per se. No, not really. No, we didn't see any differences. Yeah.
Awesome. Salu has a question in the chat, which I think is the same as what Reina has there. So asking, what was the amount of EVs that were optimized for the different conditions? So what was the dosing and how did you decide on it?
Yes. So yeah, that's something that is tricky actually. So we ended up using somewhere close to, if I'm not wrong, 15,000 EVs per cell sort of situation. So we counted the EVs and then we definitely. But we decided not based on, well, we know that in literature, people do it super in a big range, no? So there are studies that you will see like 1 million EVs per cell. But we wanted to go somewhere in the middle. We did do some preliminary screening experiments, however, not with the Alzheimer's brain-derived EVs. This is something that we did optimize beforehand, and not in the iPSC. We just tested some EVs isolated from mice brain onto the N2A cells, and then we noticed when is the concentration when you start to see the effect, and then we opted for we did some, how is it called, staining experiments also and live imaging experiments, and then we ended up deciding, okay, 15,000 per cell is actually not a bad number. So
right, so you did some kind of dose then it sees
Yeah,
stuff early on.
exactly. Yeah.
Rain is asking how you counted the EVs.
Sorry. How did we, what? Sorry.
How did you count the
Yeah.
EVs?
NTA. So NTA, of course, is not one of the best quantification tools there, but I mean, you have to do something.
Yeah, well, I know what you mean. This Alzheimer's field, it's comical how many different doses are used and what people refer to as a dose and all that. So
exactly.
It'll be interesting to see that going forth.
Yeah.
I had a question in terms of the PrPC stuff, and you've done a lot of work with the amyloid, but do you think that this would be relevant in terms of kind of other pathological protein transfer in different diseases as well?
Yeah, I would assume so. Yeah, we actually are working not that much in detail with PrPSC, but we are working there also, and there it's a little bit challenging because it's a higher biosafety level. You cannot do most of the things that we did here, and but I would assume that it would be the same for alpha-synuclein, and at least for tau or something like that. Well, tau is already published. We saw that the tau is actually packaged in the EVs. And yeah, I mean, but any kind of receptor mediated aggregation would be possible, I would assume. So, yeah.
Thank you. Are there any more questions, either in the chat or
somebody has,
otherwise?
I think, written me as a single message.
There's one more. From your observations, are SAD brain-derived EVs driven by or dependent on p-tau?
Dependent on, I'm not sure. What we did see, however, I think I didn't show in this presentation, is that SADs or sporadic Alzheimer-derived EVs have a lot of tau actually also. So definitely they are involved in somehow transport mechanism, or let's say maturation, something like this, or clumping, maybe, I'm not sure. But we did see a lot of signature from the tau protein in the extracellular vesicles. So I would not be the right person to ask this question. I'm not sure. Maybe you can talk to, who was it that published the tau story? Oh. I'm forgetting the name right now. Yeah.
I know who you're talking about as well, but I also forget who it was.
You actually know the person. That's okay.
Few people have said thank you for a great talk, and I agree, it was excellent, super easy to follow, and it was nice to to see the work that you've done. So thanks so much for spending all your time with us.
Thank you very much.
I don't know where, I guess the afternoon where you're at.
I am in Hamburg, so it is going to be afternoon. So it is happening. Thank you very much.
Enjoy the rest of your day and your weekend. Thanks
Yeah.
so much.
Thank you. Ciao, ciao.